Primary cultures of Aplysia sensory motor neurons provide a model preparation for studying synapse formation and synaptic plasticity in vitro. This video demonstrates the identification and microdissection of sensory and motor neurons from Aplysia ganglia, as well as the methods for establishing and maintaining sensory motor neurons in culture.
Hey, I'm Yali Zhao from Laboratory of Kelsey Martin at the Department of Psychiatry and the Department of Biological Chemistry at the University of California, Los Angeles.
Hi, I'm Wang, also from the Martin Lab.
Today I'm going to show you the procedure of culturing sensory motor neurons from seed slag Aplysia californica.
In our laboratory, we use this procedure to study the cell and the molecular biology of synapse formation and synaptic plasticity.
So let's get started.
To begin culturing Aplysia neurons, first prepare culture dishes by coating the glass well of a MatTek glass bottom culture dish with poly-L-lysine made in sodium borate. Add enough to completely cover the glass and incubate at room temperature overnight. After incubation, thoroughly remove the poly-L-lysine by rinsing in artificial seawater 4 to 5 times. After removing the last rinse, add 2 milliliters of a solution containing